Um, I don't know what it is, but it's a campus that just makes you feel comfortable. There's something about it, you just feel comfortable when you're here, which is a really nice praise, I think, for where you are, and I'm very pleased to be here again. So, my talk today is about technology we're using, which allows us to do unprecedented precision genetic engineering. And I've structured it, so I'll accelerate through the talk. So bear with me at the start. It's very elementary, and it's a talk I give to a variety of groups. But it gets across some of the key points, and I won't give away the punchline of what those points are in a minute, but they'll become evident as we go through the talk. So please bear with me. If you see the first few slides and you think, the phrase we have in the United Kingdom is teaching your grandmother to suck eggs. If it's that, just bear with me. Alright, so the presentation will go through in 3 sections. How and why will be the second and third section, and I intend to lead into the how, to explain what the new technologies are, put them in context basically. Right. So many of you, many people, and many of you in the room, when you think about a genetically modified animal, you're thinking along the lines of this animal here, which obviously has a bright green fluorescent snout, and most pigs don't have bright green fluorescent snout. And we've incorporated a transgene into that animal. And this is the image that the public have, that the media have, the politicians usually have. And what I want to get across today is technology has gone from here to this animal here. And I will give this punchline away. This animal is genetically engineered in the context it's been the embryo it was derived from was manipulated through the use of the genome editor. But it doesn't have a transgene in it. It has a single base deleted in its genome. So, what is DNA? This is the simple bit. Please bear with me. What is DNA? Well, we have an image of a human here made up of lots of cells. Each of those cells carries lots of chromosomes. In those chromosomes, you have these lovely long strands of DNA. Now, I still get fascinated by some of the simplistic things. Does anybody know how much, if you could stretch out all the chromosomes into one little fine thread of DNA, do you know how long it would be? Bear in mind that these cells are usually too small to see with the naked eye, and certainly the nucleus inside them is too small to see with the naked eye. You have to use a microscope. Anyone got an idea how much DNA would be? About a meter. So we package that in, and I find that absolutely fascinating. But it leads to another calculation. How many cells are in the body? No one's actually counted them, but there's approximately 10 to the 14. But let's be conservative. Let's just say 10 to the 12. And it's somewhere in that range. Most of those cells, apart from your fully differentiated red blood cells, carry a nucleus. They carry a meter of DNA. That means inside each and every one of us, we have 10 to the 12 meters of DNA. That is 10 to the 9 kilometers of DNA. That would go from here to the moon and back thousands of times. Isn't that just absolutely incredible? You package it into an organism and make it work properly. I'll get back into the topic. I find that fascinating. DNA, as you know, is transcribed into RNA, makes different proteins. Sometimes they work together, other times they work separate. This animal, with the transgene, with the green fluorescent protein derived from a jellyfish, that transgene is inserted into the DNA. So this is your strand of DNA, this is a little bit of it, and here's the new bit of DNA which we have put in. So the genome prior to this genetic modification was quite long, and after the modification it was just a little bit longer because it had that transgene in there. And the transgene might be a couple of kilobases. In some cases, it might be several hundred kilobases, even bigger. But that's not what happens in the world. That's what happens in biology. In biology, we do have the genes lined up on the genome, and we're very close to being able to accurately map all of those genes. We know where most of them lie, and in some species we know the sequence better than other ones. But they carry two individuals, or indeed within an individual, between different cells in an individual. That sequence will differ. That's single bases. And as you know, C and G go together and the A and T go. So if you change that C to an A, you've got an AT rather than a CG, as shown here. And these single nucleotide polymorphisms are just variations of the gene. And this gives you the alleles, which makes us different. That is why everyone in the room, certainly most people in the room are of Indian descent, but you're all different. Some of you are tall, some of you are short, some of you are more hair than me, some of you have the same amount of hair as me, different colours of eyes, some of you are thin, and so forth. That's because you've got these mutations dotted around your genome, giving you the allelic variation that makes us individuals. And that's what evolution plays on. The genome of most of these animals, top end here, are really quite similar. The same size, for all intents and purposes, have the same set of genes. There's some different genes. And it's the allelic variation in those genes that makes one a horse, the other one a primate, and including man. So this variation here is absolutely central to biology. It's absolutely central to life, and what I want to say, it's absolutely essential within the agricultural context, which is what I'll focus most of the talk on. So this timescale here is rather long, and if you want to do something and you have to rely on evolution, that's going to take a long time to happen. I haven't got a timeline on here, but you're well aware that evolution is a very, very slow process. A process that makes mistakes, it makes dead ends, it makes significant breakthroughs, it causes problems, it gives you disease. Mutations and variation is what drives biology, but it's also, if done by nature alone, carries risks and problems attached to it. What we can do now with these genome editors, which are molecular scissors, the nucleases, endonucleases, we can target the activity of that nuclease, which is basically a scissor that cuts DNA. We can target it to a given base in the genome. We can combine it with a template that predetermines what the sequence should be, and we can create these single base changes or multiple base changes. We can create the variation which evolution relies on. The difference is we can do it in one generation. Not meaning our generation, the generation of the animal that you're working with. Unlike evolution, which takes a long time. So the thing at the start was an animal called Pig 26, which caught the media attention in the United Kingdom a couple of years ago. This animal is slightly younger age, and for those of you with good eyesight, you'll see that there is a base missing here. And again, those of you with good eyesight will see that the sequence trace identifies that, where it breaks down from a single color into two colors. And that's what we can do, single base. And that's my single message, and I could stop the talk now and just let you all go away and imagine what you can do with that type of tool. But I'll carry on a bit more and give you some of my thoughts about it. But let me take you into the how. Just before I do that, I want to tell you about livestock breeding and what has been going on and what will go on into the future. So livestock breeding relies on inherited change. It relies on this variation that exists, single bases, multiple bases, repeats, deletions, duplications, and so forth. We know it works, and this little image here is my representation of proof. So the gene for stripe, black stripe, is present within this chap and his wife here. The difference is they have different alleles. He has a cross horizontal allele and she has the vertical allele. So therefore the son has the mixture. Has both alleles, still has the black gene, and that's what he has. You can't have diagonal because mom and dad don't have it. You can't have wavy, mom and dad don't have it. Right? It's predetermined that he'd go out on this Sunday afternoon for a walk with a shirt and a crisscross. I'll come back to that. Capturing that genetic variation works extremely well. There are a plethora of different types of dogs on the planet. Small, big, large, fat, long leg, short leg, all different types of colours, all different types of length of hair, and so forth. Big dogs like this, very ornate animals like this, tiny little ones, but they're all derived from this animal here. They're all derived quite a few millennia ago when some farmers somewhere in the world either found an injured animal or an animal that was hungry and you could look after, or pups that had lost their mother. Maybe they'd killed the mother and reared those animals and selected out the offspring, the ones that were a little bit calmer, not likely to do this to you every time you spoke to them. And from that, we've got the variation. The amount of difference between all of these, the genome difference, is actually very small. We've been doing this in livestock for millennia, about 10 to 11 millennia for that. And it's evidenced by the sort of paintings we find on caves and other demonstrations of ancient man affiliation with animals. And this is an artist's impression of an ancient farm where we've got a couple of oxen here, we've got some pigs running around and some chickens. And we've been doing it for a long time. What did we initially breed for? Exactly the same as the dog. Temperament. We didn't want these animals running amok. They might even be living in our house. We wanted them to be calm. Social structure. We wanted them to tolerate living together. So we could have a flock or a herd. And the ability to breed in captivity. We didn't want to every morning go out and find them because they'd run away. We wanted them to be comfortable that they lived with you. And we did this basically for 10,000 years. We were a little bit more sophisticated. The farmer would say, well, I like the look of that animal, it's got the right colour, it might have the right shape, it might have a straight back, whatever characteristic the farmer liked, and he'd breed from that animal. But for the first 10, 11 millennia, it was literally, I like the look of that animal, it has a very simple characteristic which I like, and I'm going to breed from it. Very sophisticated breeding sort of structures. We now breed much more elaborately, and I'll go into that in a minute, and we have production targets, milk production, meat quality, disease resistance, reproductive robustness. This would never have crossed the mind of a farmer, even a few hundred years ago. It just wouldn't have crossed the mind. The animal's alive, that's all they cared about. There might have been natural selections for disease. Within that, as outbreaks came through. It's only in the last 50, maybe, only the last few hundred years where we've been selectively breeding, and only the last 50 years where we've been able to start to apply genetics to it. We now use tools, or have used tools like marker-assisted selection, and we're now moving into genomic selection. Just to let you know the basics of that, this variation which I described at the start, single point mutation, basically it's a flag on the genome. You can use assays, PCR, or other assays to identify animals that carry that, and you can select those animals. And having identified an offspring, really good animals with strong traits, you can use your markers to go back to the parents to find out what it was. Genomic selection is coming in and allows us to go much earlier in that process to select genomically the animals we want to breed from, and we have this earlier intervention reduces the generation time and how we work out the benefit of these things, and that accelerates the genetic gain. We're now moving to whole genome sequencing, where you can, rather than having a few flags on the genome, you've got the entire DNA sequence giving you a fingerprint, a blueprint of what the animal is, and you can use that in your selection pressure. We're going beyond that into the idea of imputation. Most of the genome is exactly the same between animals, between ourselves. In fact, large tracts of our genome is exactly the same as that of bovine, or of a buffalo, or a chicken. If you can sequence a little bit of it, you can work out the probability of the bits beside it, and you can impute the sequence you don't want. So some of the more advanced projects we have at the Roslin Institute just now, rather than sequencing a genome 10 times or 30 times, we're sequencing at 0.1. What that allows us to do is, rather than sequence 10 or 20 animals, we're sequencing 500,000. This technology is trundling on at an incredible rate. And we have very clever mathematicians who can work out on calculations to give us what that genetic merit is and which animals to select from. It's a far cry from the ancient farmer who just said, I like the look of that animal. That animal has long legs. That animal has a straight back. We can apply incredible sophistication to this. Just to give you an illustration again of that, you select, in essence, the genetically elite animals, the more elite animals, and over a generation time, you shift the mean in the distribution, and over years, your yield increases. And without going into the maths, you can do it really, really well. You don't need to see the dates here. Over the last 50 years, there's been a steady increase in the amount of milk production in the dairy. This is global values. And that's reflected in an overall increase in agricultural productivity. And it's all down to genetics. Or it's not. It's down to a lot of things. But genetics is central to that. So I come back to my couple who are walking Sunday afternoon. This chap here who has the mixture of mum and dad, that's the essence of what genomic selection is. But we have a limitation. Let's say we wanted this boy to have a yellow stripe. We can't do it. We can't select that. It doesn't matter how hard we try. Dad's not got yellow. Mum's not got yellow. It doesn't exist. So we have to do one of two things. And this is one of my take-home lessons. We have to wait for evolution to give us it, a spontaneous mutation to arise in the right animal that we can select. We need luck. I don't know how many of you do the lottery, but that lottery game is extremely, extremely low chances of success. Or you can use these tools that we have, the genome editors. As I said, we can introduce that variation in one generation. So, how? I'm going to just describe a little bit of what these genome editors are. For those of you who are versed in molecular biology, you'll pick up, for those of you who are not, the take-home message is they are literally molecular scissors which will cut the DNA exactly where you want them to cut. That by itself doesn't do anything because the cell is full of machinery, whole complexes of machinery, proteins which repair DNA damage, DNA cuts, double-strand breaks. If they didn't, the cell would recognize that, it would stimulate a cascade of events which would lead to the destruction of the cell. Diseases such as cancer, which have DNA damage, get around this because they overcome and shut down that protective machinery. So we're using normal proteins that exist within the cell to help us in this tool. So the cut goes, and the two ends are cut, double strand break, and then you bring the two ends back together again. And that will keep going until a mistake is made. Because these processes, although very good, they're not infallible, and they make mistakes. And it's those mistakes that we're going to capture. So there are 3 types of genome editor that you'll have heard about. You may have heard about the zinc finger nuclease, which was really one of the first ones. There was one before that called meganuclease, and there's still some activity going on with the meganuclease, but it hasn't reached a situation which makes it attractive for regular use. There's the TALEN, which was next in the block, and the one that most of you will have heard of, the CRISPR-Cas9. I'll just go through the basics of these tools. These two rely on a restriction endonuclease called FOC1. It has no DNA specificity, so it just cuts randomly in the DNA. What the bit here, the zinc finger, or the tail, does, it gives that target. So in the case of the zinc finger, the individual zinc finger will bind a 3 base. If you put 3 zinc fingers together, it'll bind 9 bases. This restriction endonuclease is a Popplegate dimer. It has to have 2 components, and so therefore you design another zinc finger nuclease for the other strand with a specific spacer in between. So if you imagine the zinc finger of one grabbing the DNA here, the other grabs the DNA here, the 2 parts of the FOC1 come together, and you get digestion, you get nuclease activity. Remember I said that we have the DNA repair mechanism when we make mistakes. If that mistake is a small insertion or a small deletion, you can see what happens. That spacer is shifted. Either it comes too close together, or it comes apart. As soon as that happens, the genome editor will stop, the zinc finger will stop cutting the DNA, and you will have that mutation captured. In the case of the transcription activator-like effectors, the tails, they bind single bases. And you can string a whole series of them together to give you that specificity. Again, both strands because it's relying on one. And the clever bit is this and this is done in a library format. You can buy the tools for this from Adgene for about $300 or $400, and if you can work on restriction endonucleases and plasmids, within a couple of weeks, you'll be making these in the lab. These are very hard to make, and really you have to go to Sigma to buy them, and that's very expensive. They both do the same thing. This is slightly different, and the difference, there's 2 differences which make it really quite remarkable, and that is why there's a huge uptake around the world. First of all, it's an RNA-based system. It's not a protein, it's an RNA-based system. So the design, and it uses a guide RNA to give you that specificity that is given by the zinc finger or the tails. And you can design that guide RNA with an algorithm on your computer in about 3 minutes. Take your sequence, put it into the algorithm, and bang, out it comes. You then can, if you have the capabilities of making RNA yourself, you can synthesize it, or you can get a company to synthesize it and send it to you. I realize that sometimes supply reagents here in India is not quite overnight, but literally in the United Kingdom, we can design that in the morning and have the guide RNA in the lab the next morning. The endonuclease activities, a bacterial protein called Cas9, and that reflects the second half of the difference. The guide RNA is not attached to the nuclease activity. Here they're attached, they're tethered. That FOC1 is physically attached, bound to the zinc fingers, while here it's not. In this case they're not, which means that you can go in with more than one guide. You can deliver one vehicle to deliver your protein that's going to do the cutting, and you can deliver as many guides as you want in theory. So, very versatile, very easy to make, very cheap. And that's reflected a huge uptake around the globe. So just to explain a little bit about what they do, and there's a whole variety of these tools now, so I'm just giving you the simplest version. It creates a double-strand break in the DNA. This is this editor here, shown in a pair of scissors. You can now make nicking. We've got nicases, which will give you single-strand breaks, but I'll leave that for just now. That double-strand break evokes proteins. If there's not a DNA template around, it evokes proteins involved in what's called non-homologous end joining, which basically puts these two back together again. As I said, it does that until it makes a mistake. So if you cut it with the editor, it goes back together again, and it went back as normal. The editor will keep cutting it until it makes the mistake. If you put two in, and this is easier with the CRISPRs, because you can put two guides in very simply, you can delete regions to the base. If you have a template, a DNA template, plasmid or an oligo, homology-directed repair will be brought into work, and the template and the proportion events will be used to repair the mistake, and in that way you can change the base sequence of it. So literally you can delete sequences. This is just a string of ATCGs, or you can convert sequences using these tools. And you can apply it to just about everything, or as far as I know, every organism that we've tried to use these tools and it works. In the case of livestock, we work primarily with pigs and sheep, although we've got a little bit of work with cattle. These cattle were actually made in Texas, the lower cattle. It works in chicken, goats, and it works in primates. And it has been used in human blastocysts, human earlier pre-implantation embryos. And that has brought a huge debate about whether we should use these tools to engineer the human germline. And that debate carries a huge amount of ethical and societal concern attached to it. And that will run its course. But what it does do means that these technologies will be in the media on a regular occurrence. Why would you do this? So we've got this tool, and there are many wonderful molecular tools. Why would you use it? The overriding reason for using this is we currently have 7 billion people on the planet. 1 in 7 are malnourished. If we do not increase the protein production at the farm, the amount of meat, the amount of crop, then by 2050, when we have 9 billion people on the planet, 1 in 3 of us will be malnourished. Now, apart from being a morally unacceptable position, it is probably, and many countries, including India, use words like food security. Well, I would argue that if we have 1 in 3 people malnourished, it would go from food security to national security. So we have this massive ramping up of the population. And it's really quite sobering to think that I was born out here, and we're now here. What our forebears encountered in challenges are nothing compared to what we face today. On top of that, we still have diseases, although we have some successes, such as rinderpest and so forth, and a huge effort that's going on here in India for FMDV. But we still have diseases which we really can't contain. Some we can't contain at all. Some we can't treat beyond culling. And we have global warming. These numbers, 9 billion, 7 billion, to most people are unimaginable. It's very hard to get a grasp of what that means. What does it mean to me on a daily basis? Well, I took this from Wikipedia. This is images of your lovely city here, Bangalore, and that 9 billion by 2050 can be translated into the fact that in 2 months, if all the new people on the planet were put in one place, you would have another Bangalore. 2 months, and you'd have 2 Bangalores. That is quite a sobering thought. So that is the driver for us trying most of the stuff that we do on our daily basis in our research. And my pitch to you is that genome editors are one of the ways we can do this. And I'll let you quickly read this, but this is the challenge we have within agriculture. It basically says, whatever we produced food-wise in the past, we have to do it again, but we've only got 25 years to do it in, rather than 10 millennia. So that is really quite a challenge. So I want to give you one example of the project that we're working with genome editors to illustrate how you could use these reagents. And what I'm not trying to say is it will replace genomic selection. It will replace the standard genetics approaches to enhancing the economic merit of an animal. What it does is it allows you to get access to variation which you would not normally get access to. You will know of breeds of animal that carry some good trait. I do apologize, and I meant to learn this name, but I haven't learned this name. There is a cow from here in Karnataka. The last time I visited, I saw it in NDRI just across the road. And I think it's called Malnad Gida, or something like that. Close? Okay. It's supposedly very resilient to foot-and-mouth disease. That's a wonderful trait to have. But you can't take that animal and breed it easily into one of your high-producing animals, your larger animals. Because as soon as you do that cross, you have half the alleles from Malpighian and half the alleles from whatever animal you cross it into. And yes, there might be in those 20,000 genes, 20,000 alleles, the one or the few that gives FMD resilience, but you'll have a whole lot of other alleles which are not quite so good. So it gives you, these tools allow you access to that. An extreme version of that is where you identify variation in a species that has some trait attached to it, but that is not the species that you want to work with. Okay, so this is this example here. It's a disease called African swine fever, caused by African swine fever virus. This is what it does. It kills pigs. These pigs die from internal hemorrhaging within about 5 or 7 days. Grotesque destruction of the inner organs. If we have an outbreak, it's reportable to the OIE. We cull and burn. And there is, in essence, 3 types of pigs on the planet. I apologize if any of you in the audience are aficionados of pig breeds, but bear with me a second. The very ancient ones in Indonesia called the babirusa, which are the lovely tusks and the hairy. And you have African pigs, tush pig, warthog, and such like. And you have everything else. Right? And that includes all the pigs. Probably includes all the pigs that exist, all the different types of pigs that exist in India, both wild and domesticated. And certainly includes the wild boars that exist in Europe, as well as the pigs. Now that becomes important in a minute, because we need to do something for this disease. We have no vaccine, no drugs, we cull. No treatment, we cull. That's all we can do. And when outbreaks happen, that is indeed what we do. So, this virus, African swine fever virus, is carried by a soft tick. If that soft tick bites an African pig, that African pig is subclinical. It's possible that all indigenous African pigs in some areas are seroconverted to this virus. They show no symptoms. As far as we know, they show no symptoms whatsoever. If that virus infects one of the other pigs, pigs here in India, pigs in the United Kingdom, pigs in America, pigs in China, wherever they might be, they die. They die. They die very quickly. So what that tells us is there's something, the same virus, obviously, there's something in the genetics of these different pig species which is beneficial for this animal and absolutely catastrophic for this animal. So why don't we just mate, take our domestic, the same argument, to a certain extent, the same argument made for the cattle, except it's more extreme here, and that these are pig species. So these species are as close to each other as we are to marmosets. So crossbreeding is not possible. We know an awful lot about the virus, and like most viruses, it encodes a number of immune modulatory genes, and we've identified a series of those. One of them goes by the exciting name A238L, and A238L looks like a protein, a cellular protein called NF-kappa B. NF-kappa B is the cytoplasmic inhibitor of NF-kappa B activity. NF-kappa B is a transcription factor involved in most immune responses. It is absolutely central to activating cytokines, and it is a cytokine storm that leads to the dramatic hemorrhaging you see in these animals once infected with the virus. So here we have inside the virus encoded by protein which suppresses the pathway which leads to the death of the domesticated pig or the wild one. So our approach was to look at the genetics behind that. We sequence as many of the genes involved in either the NF-kappa B pathway or its associated pathway NEFAT, as many of the components as we could. We did this some years ago, but when the sequencing wasn't quite as robust as it is nowadays, and we did it in essence before the pig genome sequence was properly annotated. Most of the genes, it was a student of mine that did this project, and came back to me after a week or so and said, Bruce, I've sequenced the genes from African pigs, and I've sequenced the genes from our domestic pig, and they're exactly the same. And I would say, go away and sequence some more. And he went away, and he'd sequence some more, and he'd come back after another few weeks and say, they're still the same. And this went on for some time, and I believe the student started to question the logic behind his supervisor, until the day he hit on a gene called P65. P65 is encoded by the gene Relay. And this is just a cartoon of it, all right? So we have two versions of Relay. In the domestic pig, we have this version. In the African pig, we have 3 amino acid changes. One of those amino acid changes is one here, encodes a serine, and that is phosphorylated. And we know that in the human version of P65, that phosphorylation regulates transcriptional control of the gene. We know now, our pig versions, that this is about half as active in an in vitro assay for NF-kappa B signaling activity. The full induction of this is the same as this animal, but this protein, but it only brings it up to the basal level. So the full induction in an in vitro assay of the African version of P65 is the same as the basal level of the domestic P65. Remember, it's a cytokine storm that kills this animal. So we've used our genome editors since we can't breed these animals together. The other thing you should know is that in the intensive pig breeding industries, where there are a number of significant companies involved in the germ stock of those industries, they work with elite animals, and it's the semen from those animals that go into the production system. We have sequenced this gene in the vast majority of those nucleus herds for a number of companies, and we have not found the African variation. We believe it's lost from the domestic pig genetic repertoire. So what we're doing is we're taking our genome editors to convert those 3 amino acids. And we've done a series of animals, some that have all 3, some that miss this whole endpoint of the protein, some that just have these 2, and we're working on animals now that just have the last amino acid converter. So we have changed the allele in the domestic large white landrace animals that we have to have the allele which is present in an African. Our hope is that rather than the virus was to infect these animals, rather than ending up like this, it would end up like this, and it would trot around the pen and be quite healthy. Some of these have involved changing a few bases, 5. To get all 3 amino acids converted, we changed 5. And I can explain, if people want to know why we changed 5 rather than 3, it's to do with how the editor works, and I can explain that if anyone wants to ask afterwards. But we also have as I said, animals with a single base. So these animals are genome edited. They carry a deliberate change in their genome and their sequence, which we have engineered, but that's all they carry. There's no transgene, there's no selection marker, there's no plasmid, there's nothing else. The editors are delivered as RNA, they degrade within a few hours, they're no longer there. The only difference between this pig and its sibling that's not edited is this single base. And you could sequence as many of these animals as you like, and you'll be able to identify the base changes, but just identifying it would not tell you that I or my colleagues edited it. They could have spontaneously arisen. There's no difference between this SNP and one that you would find randomly caused by evolution. Right, and I'm just finishing this last slide to indicate this is our challenge. We have diverse farming around the world. We have a whole variety of farming structures here within India. This technology can be applicable here as it is in a massive farm in Missouri or wherever it might be. It won't replace the need to develop drugs and vaccines. It won't change the way we select our animals in agriculture. It will just give us more variation to do that selection with. And one of the reasons why I'm visiting India this time is we've just had a Department of Biotechnology meeting discussing how we could try and help accelerate the uptake of these technologies in this country. At that, I will stop. Thank you very much.